So friends, we were discussing about the natural polymers that are used for immobilization of plant cells, and we discussed about alginates. The second natural polymers that are used for immobilizing the plant cells are carrageenans. Now carrageenans are nothing but they have a backbone of alternating 1,3 linked beta D galactose and 1,4 linked alpha D galactose, but they differ in the number and position of ester sulfate group. When we say they differ in the number and position of ester sulfate group, here I would like to emphasize that different types of carrageenans are available or are obtained from the marine weed belonging to Crispus genus, and these are different variants are kappa carrageenans, iota carrageenans and lambda carrageenans, and these differ in the number and the position of the ester sulfate groups present in their structure. Now the overall process of using carrageenans is almost similar to that of the alginates, and the gelation can be achieved by cooling the solution of carrageenan or by reacting this solution with the solution containing gel inducing reagents such as potassium or ammonium solutions or calcium solution and so on and so forth. But for carrageenans, most widely used solution is of potassium chloride, and this basically also helps in hardening of the beads of the carrageenan that are formed. Now the major advantage of carrageenan over alginates is that these carrageenans are less sensitive towards the chelating agents than alginates, but alginates provide more spherical beads in comparison to carrageenans. So if the shape of the immobilized structure is not of importance, then we can also use carrageenan. But if a spherical shape is required, then alginates are the material of choice. Another natural polymer is agarose, which we know that is nothing but a purified constituent of agar. It is a linear polysaccharide that is composed of repeating agrobiose units consisting of alternating 1,3 linked beta D galactopyranose and 1,4 linked 3,6 N-hydrogalactopyranose units. Now again, the process is almost similar, except the agrobiose or agar has to be heated in order to keep them in a solution form. So once after heating, the agar or agrobiose is converted into a solution. This solution is then poured into the cell suspension. Once this mixture is prepared, this mixture has to be maintained at a temperature of 5 to 10 degrees above the gelation temperature of agar or agarose. Otherwise, this mixture will get gelatinized. So once this mixture is formed and is kept above the temperature of gelatinization, this mixture is then poured into suitable molds, and once a mold is achieved, then it can be disintegrated into spherical beads. Now these spherical beads can also be obtained through emulsification using a vegetable oil, wherein this molten mixture of agarose or agar along with the cells is dropwise put into an ice cold buffer or in a vegetable oil using a narrow orifice resonance nozzle, and we get spherical beads. Now the major drawback of using this polymer is that we need to maintain the mixture at a higher temperature or we can say above the gelatinizing temperature, and that higher temperature can sometimes damage certain type of plant cells. Now we move on to some other natural polymers that are used, and these are pectins or pectinates, gellan gum, hyaluronic acid, collagen, gelatin, which is used in the range of 10 to 20% weight by volume. But again, the problem with gelatin is the cross-linking of gelatin is largely done by reacting it with glutaraldehyde, and we know that glutaraldehyde is toxic to some of the plant cells. So one has to be very careful while using gelatin or while using glutaraldehyde as a cross-linking agent for some other polymer. Now we will discuss certain synthetic polymers that are used for the entrapment or immobilization of the plant cells. Now, as we briefly discussed that synthetic polymers has certain obvious advantages over natural polymers, and these are hydrophobicity and hydrophilicity of these polymers can always be modified by modifying their chemical structure. Since we are synthesizing these synthetic polymers, we can always play with their chemical structure in order to get the desired properties out of these polymers. Likewise, porosity of these polymer gels can also be modified. Polymer can be synthesized with the desired properties as we have discussed, and these synthetic polymers have a superior mechanical strength and longevity in comparison to the natural polymers. Polyacrylamide, which is one of the most widely used synthetic polymer for immobilization. Now, for using this polymer, first we have to polymerize the monomeric acrylamide molecules. So, we can say that entrapment is done by polymerizing or polymerization of the aqueous solution of acrylamide monomers, and for this we use certain cross-linking agents, and for acrylamide or for polymerization of monomers we use NN-methylene bisacrylamide. Now, whenever this bisacrylamide is reacted with monomer of acrylamide, it changes the configuration of the system, making it water insoluble, and the solution eventually gets converted into gel. This whole process of polymerization can also fasten up by using certain chemical catalyst, and one of the most widely used catalyst is NNN prime tetramethylene diamine, or also known as TMED. But the word of caution here is this kind of polymerization should always be carried out in absence of oxygen, because the monomers which we are using to polymerize, they have better affinity for oxygen, and they will lead to the formation of certain oxygen radicals, thereby reducing the rate of polymerization. So, one has to be very careful while performing this polymerization reaction. So, in this diagram or in the picture, you can clearly see the process of immobilization using polyacrylamide gel. So, first the monomers are reacted with bisacrylamide, which is nothing but a cross-linking agent, and to this mixture the cells are added which are supposed to be immobilized. Along with the cells we also add TMED, which is nothing but a catalyst, and eventually they will change the chemical configuration of this whole structure, leading to the formation of porous beads or the immobilized entrapped cell in polyacrylamide. There are certain disadvantages, and the most important disadvantage is that the monomers are toxic for certain cells, because we are adding the cells into the mixture of monomer and the cross-linking agents, and sometimes these monomers decrease the viability of the entrapped cells. Further, during the polymerization process, heat is also evolved, which also decreases the viability of cells. But these disadvantages can be overcome by using polyacrylamide hydrazide. Now, in this polymer, linear polyacrylamides are partially substituted by acyl hydrazide groups. Now, this polyacrylamide hydrazide, this provides a superior mechanical stability. Further, the monomers of polyacrylamide hydrazide are non-toxic to the cells, which was the major disadvantage of polyacrylamide, and the gels that are formed by using this modified polymer, they are less brittle, they are chemically stable, and these gels are not affected by change in the pH of the media or the salinity of the media. Next type of synthetic polymer is the photo cross-linkable resin pre-polymers. Earlier we were using monomers and we were subjecting those monomers towards polymerization reaction, but here we are using pre-polymers. So, we are again removing the toxicity of those monomers if there are if some monomers exhibit toxicity. So we are using pre-polymers here. Now these photo cross-linkable pre-polymers have a specific property that whenever they are exposed to near UV light, they start getting polymerized and get converted into gel. Now these pre-polymers have at their terminal end of the chains, they have certain photosensitizing agents attached, as you can see in the diagram. And because of these photosensitizing agents, whenever they are exposed to UV light, they absorb certain amount of light, get the chemical configuration changes, and they get converted into a gel. Now there are certain pre-polymers with photosensitive groups that are available in the market, and these are polyethylene glycol, dimethyl acrylate PEGM, hydrophilic ENT, hydroxyethyl acrylate along with PEG. Then we have a hydrophobic ENTP, which is nothing but isofuron diisocyanate along with polypropylene glycol. So these are some of the pre-polymers, photosensitive pre-polymers that are used for the immobilization of the cells. Now in this diagram you can see the entire process of entrapment or immobilization of the cells with the help of illumination or with the help of light, on which these photosensitive resins or pre-polymers work. So we take a mixture of plant cells along with photosensitizer or the pre-polymers, and this mixture is then poured into suitable molds and is exposed to near UV light. Then once they are exposed to near UV light, these pre-polymers, they got jellified and thereby entrapping the cells. Now apart from UV light, this gelatinization can also occur with the help of laser light, and there are certain advantages of using laser light because the laser light does not get absorbed by the cells, and therefore sometimes the absorption of UV light damages certain cells. So this kind of problem can always be overcome by using laser light because it is not absorbed by the cells until and unless exogenous cell binding chromophores are present. Then heat is also not produced by using laser light, which will again help in increasing the viability of the cells. Polymerization can proceed extremely rapidly even in oxygen containing aqueous environment at the normal physiological pH. As we discussed that some polymers react with oxygen and they reduce the rate of polymerization. So by using laser light and pre-polymers, such kind of problems can always be overcome. Other photosensitive pre-polymers that are used for immobilization are polyvinyl alcohol PVA, which is also known as PVA, polyurethane, radiation sensitive polymer, stimuli sensitive hydrogels. Now these stimuli sensitive hydrogels are also known as smart hydrogels or smart material gels because these gels change their configuration under different conditions. Under some gels at elevated temperatures, they lose their gelicity and become solution form, or they become loose gel, and when the temperature is reduced, they again get shrinked and they form a rigid gel. And some stimuli sensitive materials are such that at elevated temperatures they shrink and at low temperature they expand. So such materials are also available in the market. There are certain advantages of synthetic pre-polymers over gel forming polymers. Now the entrapment procedure while using these pre-polymers is quite simple and rapid and is done under very mild condition in comparison to the gel forming polymers. No polymerization of monomers which are toxic to certain cells is required by using these pre-polymers. The network structure of the gel can be controlled by using pre-polymers of optimal chain length. Now since we are synthesizing these pre-polymers, we can always play with their properties. There are certain material in which by increasing the chain length of the polymer, we tend to produce certain rigid, fragile or hard gels. Whereas in certain polymers, by increasing the length of chain, we tend to produce certain soft and more suitable gels, and vice versa also when we decrease the chain length. We can also manipulate the physicochemical properties of these gel by selecting suitable pre-polymers which are synthesized in advance and again in absence of biocatalyst. So in the nutshell, the crux is we can always manipulate different properties of these pre-polymer gels when we synthesize them, and this is one of the biggest advantage of the synthetic polymers. Now the second technique of entrapment of the cells by porous materials is by using preformed support material. First method was by using hydrogels, and second is by using preformed support materials. Now this kind of method uses passive or natural immobilization of cells in bioreactors or the culture environments. Now, what happens is the cells which are to be immobilized, these are inoculated in the sterile media, and these medias are then transferred into bioreactors which contain specific membranes or the support materials which prevent the movement of these cells and confine them inside the reactors. But these these preformed support materials are always porous or selectively permeable, which helps in the transferring of the desired product outside the membrane and transferring of the nutrient media inside the membrane towards the cells. In addition to the required product, various byproducts or the toxic materials that are formed because of the normal physiological function of the cells are also transported across the membrane outside the system. And this mass transfer of the products inside the membrane or outside the membrane occurs with the help of molecular diffusion or by the convection mechanism. But as the density, cellular density increases inside this system, then the convection becomes a rate limiting step. So one has to be very careful in maintaining the appropriate cellular density inside such systems. Now there are certain advantages of preformed immobilization. This method is quite simple and have a high degree of cell viability after entrapment because you are not exposing it to certain harsh conditions. The preformed matrix is resistant towards microbial attack and therefore can also be reused even after steam sterilization. These preformed matrices can take up large volume and thereby helping in low cellular density in comparison to the other methods. When we talk about take up large volume, it means these matrices take up large volume inside the system, thereby reducing the overall cellular density within the system, and hence it helps in proper transfer of the material in comparison to other methods. Now here is a list of certain preformed matrices that are commonly used, like polyethylene terephthalate, then poly-L-lactic acid, polyglycolic acid, polystyrene is there, alumina is there, ceramics are also used, silicate is also used as a preform matrix for immobilizing the cell. Third mechanism is containment behind the barrier. Till now we have discussed the immobilization on the surface and immobilization by entrapment in a hydrogel or in a preform matrix. The third mechanism is containment of the cells behind a specific barrier, and this type of method or a system is largely used when we want to separate the high molecular weight product or a specific product from the entire effluent or the biomass. Now this type of system can be of two types. First one is the microencapsulation or entrapment behind a porous membrane, or by using normal flat membranes and the preformed matrices in the form of hollow fibers. Now when we talk about membranes, then these membranes are simple or membranes having a specific porosity that are used in ultrafiltration processes. Now we will discuss the first method, that is microencapsulation or entrapment. Now, as the name microencapsulation indicates, this method also involves the formation of a spherical bead, a spherical gel mold, but the cells here are entrapped in a porous polymeric membrane. Once these the cells or the cell entrapped in gel are enclosed in a polymeric membrane, the gel matrix can always be liquefied, and then it is allowed to diffuse out from the capsule, leaving behind only the cells that are contained inside the membrane. Now, the advantages of this technique is, this technique provides us flexibility in designing the capsule. When we talk about the design of the capsule, it means the membranes which we are using to design the capsule, we have a flexibility in selecting that membrane in terms of the porosity, in terms of the size of the capsule that is formed by the membrane. Because different membranes are available that will help in forming the capsules of size ranging from 20 micrometers to 2 millimeters. Similarly, membranes are available which have a porosity range from small molecules like glucose to a very macromolecule or a large molecule like immunoglobulin G, having a molecular weight of 155,000 Daltons. Further, the process that is used for microencapsulation is quite gentle and avoid use of harsh chemicals which can affect the viability of the cells. This also helps in recycling of the cell containing phase, which is difficult in other immobilization methods. Now, this microencapsulation using membranes can be achieved by different methods. First one is phase inversion. Now, this method involves the induction of phase separation in a previously prepared homogeneous polymer solution. When a homogeneous polymer solution is prepared, to this solution specific agents are added which will cause the separation of the phase, thereby converting it into a gel and forming it a layer around the cell. Now, the different factors that will affect this process are polymer precipitation time, polymer diluent compatibility, and the concentration of the diluent that is being used. So, all these factors will affect this phase inversion as well as the porosity of the membrane that is formed because of phase inversion. Second method is polyelectrolyte conservation. Now, in this method, it's basically a physicochemical technique which is used to encapsulate the materials by forming a coating around them through electrostatic interactions between oppositely charged polymers. What happens here is, we prepare a suspension of the cells, and to this suspension we add a mixture of polyelectrolyte solution. And polyelectrolytes are nothing but these are the solutions or the electrolyte solutions which contain opposite ions. So, when we add this polyelectrolyte solution in the cell suspension, because of electrostatic attraction, they entrap the cells in a particular membrane that is formed by those electrolytes. So, this is how this polyelectrolyte conservation process is done. Third is the interfacial precipitation. Now, this involves the formation of polymer semi-permeable membrane around a hydrogel bead. So, first a hydrogel bead is prepared, and this is prepared by dispersing the plant cells in one liquid phase which is normally aqueous. To this phase, it is then emulsified in another immiscible liquid, normally oily phase. This emulsification leads to the formation of droplets. Now, once these droplets are formed, a polymer or its precursor is then added to these droplets in such a manner, at such a rate that they precipitate at the interface of the two droplets, thereby entrapping the entire droplet which entraps the cells. So in this diagram, you can clearly understand this interfacial precipitation process. We can see that there is an aqueous phase which contains the cells. To this aqueous phase, we add an oily phase which forms the droplets, and these droplets entrap these cells. So a hydrogel is sort of gel is formed. Now to these hydrogel droplets, the polymer is added which forms a membrane around these droplets, thereby micro encapsulating them. Another method of containment behind barrier is the use of membranes by using preform matrices. Here we can, as I told you, that we can use flat membranes or hollow fibers, and the major advantage is that they provide simultaneous separation of the desired product from the system, from the media. Now this usage of membrane can be done in three ways: by immobilizing the cell within the membranes. When we talk about within the membrane, we take a suitable membrane on which the cells are adsorbed, and then the ends of the membranes are closed with the help of a porous support. This entraps the cells within that particular membrane. Second is immobilization on the membrane, wherein the membrane acts as a support for biofilms. Here the multiple layer of the cells are entrapped on the membrane, and in this method there is a direct transfer or exchange of the oxygen as well as direct transfer of the nutrient from the bulk phase into the direction of the effluent. Third one is the immobilization in the cell compartment, wherein we use a membrane to form a sort of compartment in the form of hollow fibers, and here the cells are immobilized. The major advantage of using these hollow fibers is that they provide higher surface to volume ratio, thereby maintaining the cellular density within the entire system. So these are the three techniques or the methods by which we can use these preform matrices or membranes to immobilize the cells. So friends, in the next lecture we will discuss another technique, and eventually we will discuss the applications of the immobilization technique. Thank you.